Thanks, everyone. I'm so happy to be here today to start talking about ClinGen, or the Clinical Genome Resource. But I thought we'd start out with maybe a little bit of fun. Maybe people don't think it's fun, but an interactive element. So we're going to play a game called Guess This Color. So please, anyone raise their hand or shout out, what do you think this color's name is? Pink. Great. What else? Magenta. Any more? Purple. Fuchsia. Anything else? Orchid, bubblegum, right? We could go on and on and on, and we can come up with different names about this, right? Who's right? Who's wrong? When it comes to a color, it doesn't really matter. But if it comes to a human's health, it does, right? People interpret this color differently. It's based on your prior knowledge, your experience, and your bias, right? And so when it comes to looking at diagnosing individuals with rare monogenic disorders, it's really important for us to understand this. Why? Because we have this same issue right now. This is an image of ClinVar, or the Clinical Variation Resource, and you can see this is our different variant interpretations from different individuals who are submitting them, and they don't all agree. They can have pathogenic, likely pathogenic, uncertain significance, benign. When you are a person coming to diagnose an individual, which one do you actually believe? Which one's correct? Which one's accurate? And again, this is going to affect your whole downstream cycle of that patient, their diagnostics, their treatment, how they can get into clinical trials. So we're going to play this again. But this time, I'm going to show you some colors, and I want you all to raise your hand if you think it's the right color. How many think it's pink, the top one? Raise your hand. How many hot pink? How many fuchsia? How many magenta? Look around you, right? What did we just do? We just had consistency. We now have the same interpretation. And in fact, if anybody had some color coder on a QR code, we could get a hex code. We could get an ID that tells us exactly what the ratios of red to yellow to blue are for this color. So what was the difference between your first guess and second guess? Anybody? Your preference? Reference. There we go. Anything else? Exactly. I gave you data, right? I shared more data with you, and in sharing that data, I also gave you standards. I gave you certain colors, and I told you what their colors' names were. So that standard and that framework allowed us to have more consistency. So did any of you improve your answer, right? Did you get it consistent with the group? Yep. And overall, as a group, we had consistency. So this is just an example of interpretation. You all may do this in different aspects of your life. I happen to do this in all of ClinGen with variants in genes involved in disease. And so because of this issue that we had that was building and rising as we came out of the Human Genome Project over 21 years ago now, right? And we started to have more sequencing technologies and people getting sequenced, we started creating a lot of interpretations that, again, we didn't know what was going on. So ClinGen was created 10, almost 11 years ago now in 2013 to create an authoritative central resource to define the relevance of genes and variants in human health. It is funded by the NHGRI. We have 3 main U24 grants outlined here, and we also have co-funding from other institutional centers for our expert panels. And as you can see, ClinGen is really based on these 4 key goals that I'll go over for you. It's aggregate, right? We need to collect data. We need to collect data from many different places. We need case-level data. We need functional and experimental types of data. We just need to collect whatever data we can about genes that are involved in disease and those variations within those genes. We also need to curate, and in that is developing frameworks and SOPs. So just like I gave you a framework of colors, we sit and give frameworks, or we improve on frameworks that are giving out from other authoritative sources. And then we want to disseminate that knowledge so that anyone can be able to use it, especially clinicians and diagnosticians that are looking at the sequencing. But that doesn't preclude researchers like yourself who are really looking at figuring out the molecular mechanisms of certain genes and disease. And we want to make sure, too, that patients have access to this to just educate themselves and learn a little bit more. And then like anything else, we want to evaluate and approve, right? And so some of this evaluation is we just have an understanding that there's technology coming every single day and it's evolving, and so we have to be able to evolve as a resource as well, update our standards, update our specific classifications, and so on. And so the key goal, we really want to curate the clinical genome. We want to curate all genes associated with monogenic disease. We will curate a certain number of variants within those genes, but we certainly couldn't curate all of them at this point. There are definitely other things like actionability when we think of secondary findings that you get, which means can you get a reportable action on them? Like if you had a BRCA1 variant discovered and you didn't go in for that sequencing, that you would have potential downstream measures that are taken, like having mammograms. So how do we do this? So ClinGen itself creates an expert curation ecosystem. So we derive what we call these expert panels. And again, we need experts for these, not only in clinical areas, but we need it in genetics. We need them in those experimental and model organism systems. We also have biocurators. These are individuals who just really want to collect that data, read through the literature, and apply those frameworks. There's no particular expertise on that, so it really helps us to be able to train the next generation of individuals that want to be in clinical genetics, and also to help with different networking. And then obviously coordinating all of this, because as you'll see on the next slide, we have a substantially large number of individuals that work with ClinGen. ClinGen itself wants to develop, as I mentioned, those frameworks. We have those standard operating procedures. We train the individuals that are using them, and we provide other recommendations for them as well. And then we also, because we understand we need to disseminate the last part, we have to build the infrastructure to get here. So we have a whole data platform working group and modeling group that allows us to input all of this data that we have, apply those frameworks, and be able to disseminate it to you. And so this really is a global effort. We have over 2,400 individuals from 69 different countries and 941 institutes around the world, and it's continuing to grow by the day. And I will say this is largely a voluntary community, so we probably have around 100 individuals that are supported for this at some way, some level or another, for expert panels and ClinGen staff. And just if you all are interested in looking at this as we move through a little bit more talking about gene and variant curation, is that here's what you can see if you go on clinicalgenome.org. We are going to put all of our different gene disease validity, dosage, actionability, and variant pathogenicity. You can see all of the data that we have. It's completely transparent, the semi-quantitative metrics that we're applying. And so we do have several different curation activities, but I'm going to focus on 2 of them today to review with you, because I think these are really important when we think about mutational scanning and information that we're getting back. So that's going to be gene disease validity as well as variant pathogenicity. But I certainly encourage you all, if you are interested in any of the other parts, to look on clinicalgenome.org. So gene disease validity, what is it really looking at here? It's trying to evaluate the strength of evidence that a gene is involved in a disease, right? At this point, if you look on online Mendelian Inheritance in Men, OMIM, you can find that there's around 5,000 genes that are associated with monogenic disorder. That's far less than the number of genes we know we have in our whole genome. And so there's definitely work to go through to see if there's increasing numbers, but it could be just, again, a smaller fraction. So how did we do that? We developed a semi-quantitative metric, right? This means that we apply some points-based scores based on the type of evidence that we are evaluating. Now, of course, a lot of this evidence is going to be clinical evidence. It's going to be human-level evidence. What are the cases? What are their phenotypes or phenotypic features that are presenting? What, if any, is their family segregation? But another really large aspect of this is the experimental aspect. We do look at things like the biochemical function of a particular gene. We look at its protein interactions with other genes that may be involved in that disease, model organisms. It's all relating to the molecular mechanism, which we'll get into a little bit more. But again, defining this criteria helps us to provide that consistency that we all did in the beginning with the exercise, so that we can apply that equally across all clinical domains. But there's an interesting conundrum that comes on with gene disease validity. Sometimes it's a simple, straightforward answer. Here's a gene, and it's only involved in one disease. That's all you need to curate. What happens when you have this? Genes involved in multiple diseases or phenotypes. Are they really distinct? Are they really part of separate diseases? Do they have distinct mechanisms? Are they part of a spectrum of disease? This is really important for us to begin to understand. And if you look at the bottom, even if it is older, I could get a snapshot from OMIM for you today. It's going to be the same percentage. About 25% of all of the disease genes that have been noted in OMIM always have 2 or more phenotypes associated with it. Why does it matter? Well, when you're using a semi-quantitative metric, the inclusion and the exclusion of data matters for your final classification. So being able to understand the differences in the molecular mechanism, right, or how their phenotypes vary is going to be important in how we decide the gene-disease relationship. This also comes down to those types of functional assays and even down into the variant, right? We don't want to ascribe a classification on a variant for the wrong disease. We really want to make sure that we have the most accurate reflection of that disease. So ClinGen does what it likes to do. It comes out with recommendations. And so we came out with this lumping and splitting, and it was really a way for us to define the disease entity. We look at these different criteria here. So we want to respect that there are assertions that are made in the literature, but just as you all could sit here and tell me, we all know that literature is biased. It's biased to novelty. So there will be novel assertions that, yes, this is a different mechanism, or, oh, this is a new phenotype never observed before. It's really our job to say, is that accurate? We then want to really focus on the molecular mechanism. Do we see differences in the variations versus loss of function, gain of function? Do we see that many of the variants with this specific disease are located within a specific exon or a transcript? Again, when you're thinking about functional assays for anything, this is a really important element to consider. We also look at phenotypic features, which again, in an assay you may or may not be able to do, but among individuals we can see how those phenotypes are observed with individuals with different variation. And then finally, the inheritance pattern we want to respect. Oftentimes an autosomal recessive, where 2 alleles, right, a biallelic inheritance, is affected, it's loss of function. But when you have autosomal dominant or monoallelic, you can have a plethora of loss of function, gain of function, right? Those can be downstream mechanisms at the protein level and not the gene. So we have to think about all these complexities and the information we know about these. And so we defined a process called pre-curation, and I want to just point this out to you because we wanted to be really explicit to our users and anyone using this data, right? We are collecting a ton of data that we would love for society to use in data science applications. And so with this pre-curation, we definitely let you know about what are the phenotypes, right, that are included in this final curation. It may not be the name when you look at it you expected. This one's Med12-related intellectual disability. That's not something that people typically get diagnosed with, right, from their physician or clinician. But one of those other eponymously named ones may be, and you'll see that they're all actually what we consider part of a phenotypic spectrum. They aren't distinct, in fact. They are this intellectual disability syndrome. And so it's really important when we again think about defining that disease entity and the inclusion and exclusion of the data that we have in the parsing of that data because of that final classification. If I went through with any of those genes there and said, sure, I'm looking at element A and I'm going to just look at dilated cardiomyopathy and I'm going to ignore the 9 to 11 other phenotypes that are there, what I'm doing is potentially saying I have a limited classification. Now, again, I didn't do this curation, so this is just all hypothetical. If anyone in the audience, it's probably definitive. But it matters because the American College of Medical Genetics and Genomics, which is an authoritative body, has declared that looking at what genes should be on genetic testing panels should also be looked at for the final classification through ClinGen standards. So if you don't meet a moderate, strong, or definitive classification, you as a company or as a physician should not order that particular gene on your gene panel, and you certainly should not turn back any of the variants above a VUS, a variant of uncertain significance, right? And so this means that it's going to limit you with the ability to go to pathogenicity on that particular gene and have that testing. And so these classifications are important to consider when you're thinking about those diagnostic results that are being returned. And so I'm going to move on to variants. So, right, the important part I want you to get is that ClinGen has to evaluate everything on the gene level first, because we are defining the disease entity for the curation that then goes and directs us towards the actual information that we can collect. So this sets the whole stage for variant curation, right? We need to understand that gene-disease relationship to know what evidence, again, we can collect for each variant. And just as I showed you earlier, it gets confusing. I think it's great. I've heard some of you already in your talk say, I went to ClinVar and I picked the benign and I picked the pathogenic from ClinVar. How do you know they're accurate, right? When we have some of these where we have conflicting interpretations. So what is the trusted source? And again, that's where ClinGen came in to say, you know what, we really want to work to have that consistency. And when you have variants like this, and they are labeled in ClinVar as conflicting interpretations, we can really help to resolve this. And so again, we improve on, or at least build on and adapt from the American College of Medical Genetics and Genomics current standards for variant interpretation. And so most of this, while it's really good to lead a lot of diagnostic labs and how they should consider this evidence, and some of it you will see here is functional analysis, right? Predictive analysis, besides all of the human kind of what are their phenotypes, are those consistent, is that they aren't really useful for all diseases the way they are. BA1 is the benign standalone criteria. That is currently 5%. If you see a frequency of a variant in the normal or healthy control population, so we all go to NOMAD, as 5% and above, it's benign. What happens when you have a disease that is 1 to 100,000 prevalence? You will never see 5%, right? Benign for that is 1 to 50,000. Right? If I want to be more, liberal about it, I could say 1 to 10,000. But that changes and shifts. And so that's exactly what ClinGen's expert panels are doing. They're coming through each of these and adapting it for their specific gene-disease relationship. They're also going to be highlighting, or I'll highlight later, is trying to figure out what are the functional assays that are the best to be able to use for these particular adaptations. And so we go through a 4-step process to make a VSEP. Everyone goes through step 1, which is just defining the scope. What genes are you going to look at? Some groups pick one gene, right? We have the P10 VSEP. That's the only gene that they look at. We have others like our limb-girdle muscular dystrophy that are looking at 7 genes. Sometimes it's great because we can cluster genes of the same biochemical pathway that we assume has the same mechanism, et cetera. And then they're going to go through and adapt those ACMG criteria I just showed you, and then they need to pilot. We actually have to test and prove what we adapted is going to be consistent and functional. And then we get into that same sustained curation mode. So I just want to give you all a flavor of how these things, VSEPs work, right? The time that we take to do this on average is a year to 18 months getting from step 1 to 4. It can take longer. Sometimes it can take shorter. But it's a whole process of collaboration and talking with experts. What's really nice is, as I told you about those systems that we needed to do, as we've grown and we have over 115 expert panels between gene and variant now, we needed to create systems. And so we have what we call a criteria specification registry. And so basically, every one of those adapted ACMG criteria you can access from this particular site. What's really lovely about it, and I'll show you later, is you can either download all of them, so you can look and pick your favorite gene and figure out what that VSEP is doing, but you can also look across the specific codes. So that all-important PS3 functional assay, you can see all of the currently approved assays that are being used across ClinGen variant curation expert panels. And why is this process, you might say, gosh, this seems like a long process, right? An involved process. Why is that? Some of it's because that's just what we need to do to have that accuracy, but we're also FDA recognized, so that shows the rigor with which we do this particular work. And so it's very helpful when we think about in ClinVar, if you're looking at any of your favorite genes and you happen to see this FDA recognized database, you're only going to get that from ClinGen and one other one, which is a cancer space from Memorial Sloan Kettering, OncoKB. And so we are the only 2 genomic databases that are recognized for our variant interpretations. So definitely you'll see the label there. If you're looking at any of those, you know they've gone through the ClinGen process. So because of this process, we also had to create what we called an evidence repository. So again, if you're wanting to look at genes and being able to download a lot of the information about those variants and classifications, this would be that system. You can see there's over 6,800 curated variants right now. And again, these can only come from our step 4 VSEPs. This is not a compendium of all the genes that ClinGen VSEPs are looking at yet. In fact, I don't have it listed here, but it's over 350 genes that we have currently approved for our variant curation expert panels to review. And you can see why we have this process. All of you, or many of you, have gone to ClinVar. If you haven't, I encourage you to go and look at this database. It can be really nice, but there are times where you will get an interpretation or a classification and there's no data associated with it, right? Here's this variant, it's pathogenic. Okay, tell me why. Nothing's there. How frustrating is that? Right? So ClinGen, because of our level, we do show you each and every score that we have. You have an evidence summary, and you're going to see the strength at which we put that score. So we value those FAIR principles. We value the transparency of the data because we want it to be recapitulated by others. And so as you can get the picture here, I'm a downstream user. ClinGen is a downstream user of much data, right? We can have genetic data, certainly, right? We are called the clinical genome resource. We care about people and clinical genetics. But we also understand that we have to be on a level of functional, right? We need to understand those functional assays. We need to understand the mechanism. And we also need to understand some of these computational and predictive aspects. And when we have all of this kind of data together, that means we can get to something like pathogenic, right? This is where we want to go. We can be able to understand. Or in the converse, we want to know it's benign. We want either end of that spectrum of pathogenicity. But what we also know is if we're missing any of this data, we're going to end up with this variant of uncertain significance. We've got millions of them, right? And that's super frustrating. If you put yourself in the shoes of, I've just been diagnosed, I know I have a disease, I know that it is genetic in its etiology, but I just got returned, we don't know this variant that we think could be it is really it, right? Your odyssey continues. You could get treated, but are you going to be able to go to clinical trials for gene therapy, pharmacogenomics, and drugs? No, you won't. And so what do we need? So some of this is a ploy from me to you, right, from ClinGen is, of course we need clinical data, but for this audience, we need well-validated functional assays, right? We need it documented and readily accessible for us to have this information about pathogenicity. And we need, when we say well-validated, it to include known benign and known pathogenic controls, because at that point, we can really gauge and use that information. And if we can get it computationally so we can bring it into our interfaces, even better. So how do you determine a well-validated assay? ClinGen decided to put out some information about what we, ClinGen, considers a well-validated assay. This is published from Burnich et al. In 2019, and so just to take you through it, defining the disease mechanism, that can be a long amount of work and can be a lot of time. We understand how valuable those assays are, but if we know that disease mechanism, great. Even if you don't, if you have an assay, though, that has come through to be able to evaluate, right, positive and negative controls, have you included known benign, known pathogenic? We're moving from an area to where we can't use it if you didn't do that. Even if you think it's great and you did GFP and you did wild type controls and all of that, that's not to say your assay is bad. It just won't work for us, right? And so just having those basics is going to get us to supporting. That's great, but supporting is often not going to move us from VUS into the likely path, likely benign or path and benign areas. What we really need to have is those known pathogenic variants. If you have those up to 11 of them, right, and 11 combined, we can then start moving into what is strong, moderate, and very strong levels of evidence. Now, once you get into moderate, but certainly strong and very strong evidence strength, then we can move something that may have only been observed once in one patient with a phenotype and a well-validated functional assay result into that likely pathogenic area. And so again, if you wanted to look at what those functional assays are being used across the ClinGen variant curation expert panels, you can go to that criteria specification registry. When you click on the views in the upper corner, which hopefully is this going to work, views, and go to criteria code summary, All you can do is filter by code and put PS3, and while you see the first instance back of LDLR, and you can see they can go up to strong and what gets them there, you can scroll down that whole list and you can download it in a JSON file and a TSV file, and you can see how it is applied across all of the VSEPs that are currently Step 4 approved. And so what information are we asking for? I just want to point out this is the summary of that evidence. Again, we're going to ask you what the assay is, what the materials were used, what were your variants, those known benign and those known controls. What is your readout type, right? We need this kind of information to be able to understand and validate it. We want to know the replicates and so on. And so this is what MaveDB essentially used as the basis of what they were asking people to submit, because as a downstream user, we went to them going, you have such an awesome resource. We just need a little bit more data for it to be really valuable for us to use and move these variants out of that VUS range there. And again, we don't need any assay to be perfect, right? But it does need to have those controls, and it does need to be calibrated. That's what we can use. We love reading papers, we love looking at stuff, but it's so hard at the end of the day when we read it and go, this is awesome, and I can't use it. I'd really like to, and I can't, because it doesn't have the amount of data that we need to be able to evaluate it. And so one thing you may have seen in looking at all of ClinGen's work that we have, or maybe not, but we like to use identifiers, right? I plugged up the hex code for the ID. We use many of them, the OMIM IDs, Mondo IDs, HGNC, human phenotype ontologies. But we also use variant IDs. And so even beginning to know your variant in an ID helps us to bring and import information and integrate it into our systems. And so I just wanted to be able to let you all know, because you're doing tons of variants, right, on assays. Think about registering that variant. We have a ClinGen allele registry. We have over 2.5 billion. That is why it is highlighted, billion variants registered. It doesn't require you to put a classification like ClinVar at all. All you have to do is put in a transcript and see if it's already registered or not. We harmonize these, as you can see, against all the genome assemblies that we have at this point, so we make sure that we're not registering the same variant twice. We've now moved on to also doing copy number variations. So for any of you who want to move beyond the SNPs, right, and single nucleotide polymorphisms into the copy number variations and deletions, you have the ability to register those too. And so again, if you want to get an idea about the genes that are currently being evaluated between this criteria specification registry or making your way to ClinGen, you can go into our webpage, and under curated genes, you can say all curated genes and sort by the variant, and you'll see all the approved. You also have the ability to download these from our website coming into these. Sorry, this pointer's kind of wonky. But this is just a flavor of the Step 4. Again, we have 350 genes that are currently within scope of our VSEPs, and we're working to be able to get those on our website so people can see what's in progress, not just approved. And of course, I'm always happy if you want to email ClinGen@clinicalgenome.org and ask your question, then we can direct it to anyone or myself for that type of information. And so I just want to thank you all for your time. I know slightly different assay, but I hope you found it useful as a downstream user of all of your potential data. We think it's wonderful and great. It's very exciting and it's very relevant. We're just hopefully bringing in a different aspect for you to think of when you do these assays.
That was a fantastic talk. I guess as people raise their hands or want to come down the mic, we'll do a microphone runners here. I have a first question for you. In a situation where a new assay is published after VSEP rules are finalized, how can or should authors communicate a new assay to the VSEP?
That's a really good question. I would definitely say communicating, first and foremost, the easiest way is going to this email, ClinGen@clinicalgenome.org. We have people monitoring that, several people, and they can direct it to the right expert panels. But it does bring up the other issue of, right, we could publish something and tomorrow something new comes. And so just to let you know, again, in those evaluations and improve, we have standards for recuration and doing these specifications again. So it's at least every 2 to 3 years that they need to be updated. That is to, again, appreciate the fact that something could come out and we want to be able to jump on that. Now, any expert panel, though, if you reach out to them to say, there's these really great assays, I've done thousands of variants for this gene, they may decide okay, tomorrow we're going to work on this and we're going to do this next version. So everything we're doing in now is also versions, so you can see those different versions of specifications with the actual variant classifications attached.
We have a question here.
Okay.
Thank you. That was great. I had a question about strengths and cutoffs for the PS3 assays. So if you look at that Burnich paper that you showed, you're supposed to calculate this odds path score. And that works great if you have like a well-studied gene like BRCA1 or something.
Yeah.
But if you have a relatively new gene, then there's no gold standard to calculate that by. And you go to ClinGen, and all the benign variants are underreported. So do you have any advice to get out of that trap and get your new functional assay usable?
It is hard, right? Because we were ultimately saying this is a gold standard when you have information. But obviously, as you mentioned, when there's new genes and we have to move through, that's where we may not get to very strong, right? But can we get to moderate? Can we get to strong? And as we get more information, especially with data sharing across individuals, then we can maybe get more benign. Other times they'll start going through because that VSEP has started to define their cutoff criteria for benign. You can start to see in NOMAD what would be benign variants to be able to use even if they're not in ClinVar reported, right? So again, just trying to emphasize, you might not be able to get to this odds of path until there's enough information accumulated, right, across the individuals, their phenotypes, everything. But if having the data about knowing those domains, knowing the types of variations in that mechanism, you can at least get to more than supporting, right, which is going to be helpful. So hopefully it becomes a feedback loop.
Great, thanks.
Stefan Kiro, Flare Therapeutics. First of all, one comment. I think this is a fantastic resource. It's great that you're doing that, and it actually goes beyond just the clinical decisions. Also helps as a reference set in itself. Training your algorithms and whatnot. I have a question around the process of retracting evidence, actually, or removing. For example, right now you have something like BRAF V600M, which almost certainly is not real. But that's what the publications say. But is there a way to point to evidence that's most likely incorrect for whatever reason and remove that?
So just so I understand too and clarify, you're asking, is there a way for us when we think of like retractions or any type of data that is not has come back as not being valuable? We've dealt with this a little bit. I have personally as well. Went to a paper, I was like, it's retracted and it was used in a dosage sensitivity curation. It's a little bit harder. We try our best, right, to keep up with these things, but it would require on our systems to be able to have notifications, right, to say this is retracted. However, while that seems nice, and if journals can apply that, not everything is being retracted from the journal itself. So the other example we have is a really big case control study in intellectual disability and autism. So some of you had already reported on this, right? Thousands of individuals that are looked at with a phenotype that's very heterogeneous. And I started questioning this whole paper. I don't believe it. I don't believe the statistics. Things aren't coming out. And it happened to be lucky. We had an expert panel, went back to the expert panel, and one of the experts there was like, there is a bioarchive paper from this journal that says all of the statistics they did was wrong, and they corrected it. So it's only because that person knew in a pre-published journal something had been published that what in fact we were seeing and questioning had been retracted, but it was never officially retracted through the paper. So this is a situation where I got advice from someone like, if you know it's bad, go to the journal and make them retract it themselves. But it's a hard balance. We do go back to try to look at data. It would require a lot of notifications and people actively saying, I'm sorry, I did the wrong data, right?
Yeah. One thing is, though, maybe just flag all the MNVs that are currently in the database, because a lot of them are probably not real.
Yeah, and we put in place other systems, so not for papers and things, but like we use Mondo, the Monarch Disease Ontology. They have obsoleted some of the diseases we've used, and we will clearly tell you this is obsolete, and we'll change the name to it. But we are trying to do that process now where we're at least very explicit. We know something has changed, and we're doing our best to update it as we can.
Thank you.
We have one question. Hi. So we were looking at some of this stuff about gene-disease relationships, and I was wondering if, for a research group that maybe they know what the assay readout is, they have an idea of what biochemical or biological mechanism is associated with that or is being measured by that readout, but maybe aren't able to make that jump to what OMIM diseases or Mondo diseases are associated with that. Do you have any advice for how to help people in the research space bridge that gap?
Yeah, that's, I guess, always an issue that we have, right? So how can we both be able to reciprocate from individuals doing experiments and ClinGen when you can't parse what the disease entity is? And so again, I'd certainly say if you have questions, to email to ClinGen, right? We can get to those expert panel members and have those types of discussions. In fact, we'd encourage, right, more people like yourselves to be a part of ClinGen on these expert panels. It's a very valuable contribution. Oftentimes these are clinicians and they may not understand these types of assays. And I think that kind of work and collaboration would be really helpful for us moving forward, for sure. On other ways, definitely we have those pre-curations, but they're all in a process. There could be a gene that hasn't quite been curated. You can nominate it, right? You can say, look, I have all these assays. I really would like to know this information. Expert panels have expedited other gene curations and then collaborated back with the individuals who have requested those genes to be curated. We all try to do it in a process that can be timely, but again, recognizing that we're only so many people and volunteer work. So you could just perhaps be asked to join if you send the email and the request.
Okay, I think we have maybe time for one more question. Just remember to please introduce yourself before you ask a question.
Okay. Sven Diederich, German Cancer Consortium, University of Freiburg. One question about the odds path calculations that you are presenting here. Would ClinGen accept odds path calculations that are not based on the truth set of ClinGen itself? So, for example, if we are thinking about cancer genes, clinical knowledge base has a lot of positive and negative, benign and pathogenic mutations. Would you accept that?
Yes. If there's published already odds of path from different resources, we will definitely go with those. We only come in with some different when those are lacking, right? Sometimes we can have a lot of data and then no one has done a large, study to give that. So we give the guidance of, you could do the odds paths yourself, here's how. But if it's already, within a resource, it's published, we will recognize that should be accurate. If we don't, we're usually ones to go, okay, can we ask you about this odds of path? Here's some different questions that we have for you.
Okay, thank you so much.